In the name of Almighty God, I'm Masa Sankoshi, and today I want to talk about the natural and clinical history of plague from the ancient pandemic to the modern insight. It was written by Anthony Benassar and the Microbiology Department, the Biology University de l'Isla Belarus, and the Faculty of Medicine, University of the Belarus. So I want to go to the abstract. The human pathogen Yersinia pestis is responsible for bubonic. It's such a cucumber. Septicemic and pneumonic plague, a deeply comprehensive overview of its historical context, bacteriological characteristics, genomic analysis based on an ancient DNA, and modern stains, and its impacts on historical and actual human populations is explored. The results from multiple studies have been synthesized to investigate the origins of plague, its transmission and effects on different populations. Additionally, molecular interactions of why Pettis from its evolutionary origins to its adaptation to vector-borne transmission and its impact on human and wild populations are considered the characteristics combination of DNA patterns, which plays a decisive role in the reconstruction and analysis of ancient genome. Mass are reviewed. Bioinformatics is fundamental in identifying specific Y-petist lineages, and automated pipelines are among the valuable tools in implementing such studies. Plague, which remains among human stories most lethal infectious disease, but also other zoonotic disease, requires the continuous investigation of plague topics. This can be achieved by improving molecular and genetic screening of animal population, identifying ecological and social determinants of outbreaks, increasing interdisciplinary collaboration among scientists and public health care providers, and continued research into the characterization, diagnosis, and treatment of this disease. The keyword is plague, Yersinia pestis, ancient DNA, and bioinformatics. Now I want to go through the introduction. The bacterium responsible for the plague, Yersinia pestis, has profoundly impacted human story, emerging as one of the most destructive diseases responsible for several pandemic and millions of deaths. This infectious disease has caused the greatest terror worldwide, particularly to the three major plague pandemic. The Book of Samuel in the Old Testament of the Bible, written during the 630 540 BCE, in an ancient episode from chapter 5, describes the capture of the Ark of the Covenant by the Philistines and an epidemic of tumors and deaths occurring in the city of Ashdod. Several painters have been inspired by this biblical scene. Also, probably one of the best worldwide now is the plague at the Ashdod 1630 Louvre Museum, Paris, France, by Nicolas Poussin. This famous painting depicts rats running between buildings and dead and dying bodies, in theory, in the midst of the plague. Although some others have questioned the relationship between the plague described in the biblical work and the bubonic plague, fossilized remains of the fleas, Physalia, Chopis, and black rats (Rattus) have been found in the Egyptian Nile Valley, indicating that these plague carriers were already in the Middle East in 1350 BCE. A comprehensive review of the natural and clinical story of the plague caused by Y pestis needs to cover various aspects that, together with the main structure of the review, are next briefly described and afterwards detailed through each section of the present review. Effectively, initially a detailed examination of history of plague pandemics, tracing the impact of the disease from ancient times to the present, is analyzed in the introduction section. The ancient DNA studies section aims to review the presence of Y pestis in historical population and actual molecular diagnosis, genomic and bioinformatic approach to help exploring the pathogen's evolutionary history, comparing ancient with modern strains described under the next section related to evolutionary origins. A microorganism profile section follows to review the advanced produced in its characterization from the discovery of the bubonic plague agent to the actual nomenclature change, pathogenesis, virulence factors, and adaptation. The epidemiology section covers information related to the analysis of the distribution determinants and control of electoral history, focusing on patterns of transmission and the public health implications. A section on the diagnosis, treatment, and prevention of plague outlines the methods for identifying Y. pestis infections, the antibiotic treatment highlights the importance of the public health measurements such as rodent control and prevention of plague outbreaks. It also addresses the utility of point of care testing in areas lacking advanced laboratory facilities. In the prevention context, the vaccine subject is reviewed in the next section to evaluate recent advancement in plague vaccine, including oral, bacteriophage, and recombinant vaccines, and to discuss the ongoing challenges in vaccine development and the need for further research in this field. Finally, the things to do in plague research section aims to identify gaps in current knowledge and suggest future research direction to better understand and combat plague. The section 1.1, the first pandemic, the plague of Justinian. The first described pandemic, known the Plague of the Justinian, occurred in the 6th century, year 541, in the Egyptian port of Pelusium. According to the most plausible hypothesis, and continued in 14 to 21 additional waves until 715 to 767, collectively referred as the first pandemic. The first wave was named the Plague of Justinian after the emperor of the Roman Empire of the Orient, Justinian I, and it was between 527 until 565 and initially spread around the Mediterranean basin between 541 and 544. Once this pandemic finished in the mid 700 centuries, a considerable percentage of the population of these countries has succumbed. I think that is they died to the plague. Although the first pandemic has traditionally been thought to be responsible for tens of millions of deaths, the exact number of deaths remains a subject of discussion, and some studies suggest that the mortality estimates have been exaggerated and are not supported by the existing evidence. Now I want to go through the next section, 1.2, the second pandemic, Black Death. The second pandemic in the 14th century, which started in the 1320s, again reached catastrophic proportions, perhaps due to a vicious cycle of deteriorating social conditions, which caused rats to approach people. This pandemic, known as the Black Death, due to the intense cyanosis of the dying, kill approximately 1 per 4 of the European population and spread to the Middle East and the Far East. With improvements in living conditions, the disease receded in Europe, but SARS epidemic emerged in other countries. The Black Death in the 14th century AD, 1346 1353, plague caused by Y. pestis it stands at the most widespread and lethal pandemics of this bacterium in human history, causing more than 50 million deaths in Europe, according to the WHO, World Health Organization, or 25 million, maybe 30% to 40% of the population, according to Glatter and Finkelman. This is an example of those data and the precise circumstances leading to the introduction of Y pestis into human populations, causing the second plague pandemic, and the specific time and location of its emergence are not definitely established. However, various hypotheses have been proposed to explore these events, although the exact origin is still unknown regarding the original geographic source of the Black Death. It seems that it emerged in the 14th century in the Eurasian steppes ranging from the West European to Eastern Asia. This is supported by a synthesis of archaeological, historical, and ancient genomic DNA data in Central Asia. This was demonstrated by ancient DNA data retrieved from seven individuals exhumed from two cemeteries near Issyk-Kul in Kyrgyzstan, two of which were complete genomes ancient Y pestis. By contrast, analysis of historical, genetic, and ecological evidence has led to the hypothesis that the emergence of Y pestis occurred over a century before the onset of the Black Death. The suggested model posits that this early diversification was influenced by human interactions and correlated with the territorial expansions of the Mongol Empire across Eurasia, ultimately extending to the Black Sea. In the early 13th century, Italian merchants then brought it to the Mediterranean, arriving to the Messina in Cilicia via Genossus ships carrying flea-laden rats in October 1347, and from there the plague spread to almost every corner of Europe, the Middle East, and North Africa. In Europe, the plague continued its way around the west, first to France and Spain by 1348, and then reaching Germany, Switzerland, and Austria. And in the 1349, it reached Scandinavia via North England, moving into 1350, traveling along the Baltic coast. It arrived in Russia by 1351, while some regions in Europe remained untouched by the plague for decades, others experienced a delayed impact. For example, Iceland was unaffected until 1402, at which point the plague caused a substantial decline in its population, with some others estimating a mortality rate of 50 to 60% of the population within just 2 years. The impact of the Black Death in Europe was deep, and the precise death tolls remain uncertain about the exact numbers due to contradictions between different sources. In some of them, the death tolls even surpassed the total population estimates. At the time, the pandemic, estimated to have lasted 8 years, is assumed to have resulted in the demise of up to 60% of the Western European population. This resulted in notable demographic and socioeconomic repercussions across all impacted regions, with Europe historical documentation standing out as the most deeply investigated repository to date. The effects were particularly severe in major harbor cities and urban areas across Europe, leading to the devastating consequences during the epidemic's peak in cities like Venice, Florence, and Siena. Continuing to explore the dynamic of the disease, plague persisted in Europe for more than 5 centuries following the Black Death, altitude changing its dynamic by the 17th century, contributing to the gradual decline in West European over the ensuing two countries' centuries. Interestingly, a significant shift in plague dynamics occurred around the mid-17th century, with merely one reported continental-scale epidemic in Western Europe during the early 1700s. Also, improved public health measures and medical understanding help to prevent large-scale occurrences of the disease. Localized outbreaks of plague still occurred in Europe until the early 19th century, like the bubonic, which is a cucumber-shaped plague that reached to Malta from Alexandria on 29th of March 1813 and Mallorca 1820. The bubonic, which is very similar to cucumber plague in Malacca in 1820, is known as the latest plague in Europe of the 19th century. And the east of Mallorca was affected by devastating outbreak of plague that apparently arrived by sea and landed merchandise on the coast of Sanseria. The first victim on May 9 in Sanseviera was followed by up to 2,419 dead, 33% of the total of inhabitants before plague in the affected areas Sanseria, Arta, and Capdepera. So you are watching the first figure. Yeah, figure one shows the chauffeur of Sansevier in 1820 an outbreak of plague occurred in the eastern part of the island Majorca, resulting in significant mortality and demographic effects in the area. Physicians' activity investigated the entry and transmission of the disease at the time. The events inspired imaginative literary interpretations that, while diverging from the reality, contributed to the emergence of the legend. Interestingly, this legend which is now widely accepted as factual, revolves around a mysterious figure, an unmanned young shepherd. According to the legend, this enigmatic personage supposedly contracted the plague by handling a calf abandoned in the tomb, where the body of the subject who succumbed to the plague disease would have been buried without the knowledge or approval of the local authorities. The supported plague hypothetical starting point and source of the epidemic would have been disembarked from a ship without an identified name, with origin attributed from the Tangier. Chapter 1.3, the third plague pandemic. The third plague pandemic, which began in China in the 1850, was a historic turning point in our knowledge of the associated pathogen. Initially confined to Yunnan, socioeconomic factors such as the Pantay rebellion triggered a diaspora of refugees, spreading the disease throughout China. Subsequently, British merchants transported the pathogen from the Hong Kong to the other empire territories, with India suffering the highest losses due to the plague. During this pandemic, scientific advancements in Hong Kong led to the identification of the plague agent, the Yersinia pestis bacteria, and the elucidation of the relationship between rats, fleas, and disease transmission. The pandemic persisted across all inhabited continents until the second half of the 20th century, although the plague declined worldwide in the first half, with occasional outbreak occurring in Asia and Africa and sporadic cases in South America and the American Southwest. Since 1960, there has been a gradual increase with recent resurgence in 1972 and till 1975 on several continents. A total of 2,137 cases were reported worldwide in 1974, including 8 in the United States, with 164 deaths. Morbidity and mortality weekly report from the CDC 1976 and 1977. 18 cases were reported in the United States. The contemporary occurrences of plague outbreaks were of the 3,248 cases and 584 deaths reported globally between 2010 and 2015. In August 2015, two unrelated teenagers visiting Yosemite National Park in California contracted plague, separated incident likely from infected squirrels. Also, local bears exhibit antibodies against wild pests. A death case from bubonic plague in July 2020 of a teenage male in Mongolia after consuming an infected marmot was reported, whereas current plague outbreaks are manageable with antibiotics. The inherent presence of Y pests in populations of wild rodents across various regions, including Eastern Europe, Asia, Africa, and Americas, makes impossible its complete eradication from nature. During ancient time, Y pestis was a causative agent of extensive pandemic of plague, often following the pathways of human migration. The dissemination of plague was intricately linked to various human activities, including maritime trade and tractions along the Silk Road, facilitating the transportation of fleas associated with leaves, rodents, or commodities that contributed to the spread of the plague. Nevertheless, in contemporary times, occurrences of human plague cases are predominantly confined to localized epidemic, commonly termed as plague foci. Natural plague foci exhibit a wide distribution across or Asia, Africa, and the Greater America region. From 2001 onwards, the World Health Organization (WHO) has documented 40 major outbreaks, primarily originating from Africa and Asia. You can look at the link who.int slash news dash rooms slash fact dash sheets slash detail slash play access on to January 2024. Consequently, Y pestis is still a public health threat today, causing sporadic infections and occasional local epidemics. Ancient DNA (ADN) studies. The main objective of studying Y pestis through ancient DNA is to confirm its presence in historical population and provide a direct molecular diagnosis of ancient septic plague epidemic. The study of Y pestis DNA allows for the investigation of the evolutionary story of the pathogen. There are also historical pandemics and genetic changes that have occurred over time. This kind of research aims to identify specific genetic variations, which involves identifying distinct Y pestis clones responsible for these epidemics, enhancing understanding of how these devastating events spread across the populations. The assessment of the Y pestis evolutionary patterns includes comparing ancient strains with modern ones, providing insights into the pathogen adaptation and evolution over the centuries. This analysis, beyond historical perspective, emphasizes the examination of genetic makeup in ancient and contemporary strains, allowing scientists to trace the development of the bacterium and understand how it has adapted to changing environment and hosts throughout millennia. The studies based on ADNA and specifically on the pathogen egret pestis are constantly refining a number of critical aspects to enhance the reliability of results. These include the same preparation, DNA extraction, amplification, library preparation, and analysis of DNA, which is frequently fragmented and chemically altered. Given the challenge posed by the degradation and modification of DNA over time, refining these methods is crucial for ensuring the accuracy of research findings and addressing contamination concerns in study involving ancient materials. Although various methods and protocols exist, the use of validated assays, avoiding contamination, and assessing DNA preservation are crucial. A general consensus protocol for the detection of Y pestis in DNA could be envisioned following a series of steps that ensure the authenticity and lack of contamination of the DNA samples. An optimized sample collection consists of obtaining skeletals, remains from pandemic sites, or other relevant archaeological contexts, preferably teeth or bones with high preservation potential. Most such studies focus on obtaining direct evidence of past septicemic infections by identifying E. pestis DNA preserved in the dental pulp of human remains. Dental pulp is a particularly reliable source for such studies due to its natural sterile environment and its ability to remain intact over long periods in order to prevent contamination. Strict aseptic techniques during sample collection and preparation should be followed to prevent modern DNA contamination using dedicated ancient DNA facilities if possible, depending on if the dental pulp or bone samples are used. Established protocols for the extraction of ADN exist. The dental pulp samples are usually prepared by sectioning teeth at the dentin enamel junction and using rounded dental drills bits to introduce powder, which is used for DNA extractions following optimized protocol for recovering short fragments of DNA. For bone samples, the protocols include the removal of surface contaminants and the pulverization of bone under the sterile condition. Quantitative PCR assays on specific genes are usually employed for the initial screening of A grade pestis DNA. Next, target enrichment approaches are employed to capture and sequence via next-generation sequencing NGS specific fragments of Y pestis DNA. Finally, various genomic analyses can be performed by billions of sequence reads, including full plasmid reconstruction of virulence-associated plasmids like PCP1. This enables a deeper understanding of the pathogen's virulence factors and historical strength. Additionally, the detection of single new nucleotide polymorphisms (SNPs) facilitates the assessment of the genetic diversity and evolutionary history, aiding in the identification of the specific pestis lineage. Genome scanning is also employed to identify genomic regions of interest such as deletions affecting virulence factors. For this purpose, bioinformatics plays a decisive role in the analysis and reconstruction of a DNA due to the unique characterization of a DNA such as the low endogen due to damage, and specialized bioinformatics methods are required to successfully analyze the DNA. The application of standard alignment methods with their default parameters is often not suitable for DNA, requiring the development of the tailored approach to address these challenges effectively. The computational reconstruction of ancient genome from high throughput sequencing data typically involves short read alignment methods like BWA, along with standard analysis toolboxes such as SAM tools or other genome analysis toolkit GATK. However, the default parameters of modern alignment methods may not be suitable for a DNA, require adjustment to account for the specific damage patterns, which is crucial steps in authenticating the ancient origin of the DNA samples and common fragmentation in ancient samples. Bioinformatic tools and pipelines such as Polomics, Eager, and Hoops have been specifically designed to process and analyze DNA data, enabling researchers to authenticate ancient samples, identify species, and reconstruct the genomic sequence from the past. Therefore, bioinformatics is not only important but essential for the accurate analysis and interpretation of DNA data. The Polomoics pipeline offers toolkits for mapping reads, genotyping, and taxonomics, as well as a genomics profiling of DNA samples. Polometrics is designed to be user-friendly and is capable of handling the complexities and challenges associated with DNA datasets such as contamination, short fragment lengths, and post-mortem DNA damage. Another comprehensive pipeline designed for DNA data is eager efficient ancient genome reconstruction, which includes modules for pre-processing, read mapping, damage pattern analysis, genotyping, usage guide, and damage controlling and contamination estimation. Eager provides a graphical user interface that simplifies the configuration of the analysis and spreading and breakthrough to population, make it accessible to users who may not be familiar with the command line tools. Eager also incorporates new tools for efficient adapter clipping, read merging, and the duplication, which are essential steps in the processing of a DNA sequence data. The pipeline code is available in github.com /np-core eager access until January 2024 and archived with Zenodo, including the last up-to-date version 2.4, 2.5, published in November 2023 under the DOI chain 5281 Zenodo, and another version number is 1,465,61. Access on 100 1,465,061 access on 2nd January 2021. Another valuable tool in the field of the paleogenomics and ancient disease research in hopes heuristic operational pipeline system and automated screening pipeline designed for the analysis of DNA, particularly for detecting and authenticating pathogen DNA in archaeological remains. It is tailored to address the challenge of identifying ancient pathogens awaiting a metagenomics dataset. Hope stands out for its specificity and sensitivity, being able to detect pathogen DNA even when it is present in low abundance. It is also includes an evaluation of DNA damage patterns. Thank you so much for watching my tutorial video until then. Today is 22nd of June 2026. Time is 3:14 p.m. If there is any simulation for this article literature, you can find it from github.com/mahsul script or Koshi script or Safra script.